Okay, so welcome to freshwater cyanohab monitoring training for the Phytoplankton Monitoring Network. So what we're going to be doing is going through a bit of the background of the program, some background information on harmful algal blooms caused by cyanobacteria, why we are interested in monitoring for them, and having you involved in that process, of course. And then we'll be going through what you're actually looking for under the microscope. That's kind of the important part as far as logistics and details go. So what our program is about, we are a national volunteer organization that monitors for potential harmful algal blooms. And what we do is we train citizen scientists, no matter what their scientific background may be, on how to collect samples from either a coastal environment or, in this case, freshwater environments. We're going to learn how to identify those organisms that could be potentially harmful. And you will then enter what you find in your water sample into our database. Now, what do we do with that? We will then have a better understanding of what's present in your body of water. And based on what you find and how much you find, we will then potentially be able to analyze that sample. Now, that ability is pretty restricted due to budget cuts, but we will talk about that more in the protocol section. And based on what you find, we will then alert the state or local agency to the presence of that bloom, because we know that a lot of agencies aren't able to monitor every body of water. So by monitoring those that they can't, we're increasing their awareness and knowledge of what's going on as well. And long term, what we're trying to do is identify trends with time and how these HABs might be changing in their geographical distribution as well. And I see we have a question from Sam. You can't see the slides, you're only seeing the chat. Oh, well, that's not good at all. Let me unshare then for a second. Gotcha. Well, raising your hand was what caught me onto it, so hang on one second. Oh, you know why? Because I made this chat window so big, I think. Maybe? All right, hang on, I'm gonna start over. First monitor, share. Now can you see the slide? Okay, there we go. Excellent. Okay, great. So that's the slide that went along with everything I just said. So you didn't miss any other slides in case you were wondering. Wonderful. Technology. So moving on, we, over the course of the existence of PMN, we have had groups and participants from a wide variety of backgrounds. We've had school groups, civic groups, aquarium groups, other types of educational groups. We work closely with tribal communities and just regular interested individuals. So the orange dots you see on the map are sites that have or currently exist within our original program, which is the Coastal Monitoring Program. And the green dots, of course, are our newer freshwater program sites. So we're going to have a lot more dots to add to the freshwater site after this training. You will be provided or have already been provided with the equipment that you will need. So we're sending you everything you're going to need to go out to your site and sample. So you're going to get a thermometer, gridded slides, cover slips, bottles that you'll use to collect the samples, as you see these gentlemen doing here. A Lugol's preservative, so that's how you can preserve your samples long term in the event that we ask you to send them to us. This will be very closely coordinated, so we'll talk about that process shortly. But you will be receiving a prepaid overnight UPS label in the event that we do ask you to ship that sample to us. I did already send, hopefully, your identification sheets for the target species. So there should be a one-pager that has all of your target organisms on it. You should have that handy for the Mentimeter portion at the end. You can either have it printed or open that file in a new browser tab as well. If you haven't yet received it, please type in the chat box and I will make sure that you are on my mailing list. So the training is going to cover, again, the background of HABs, the protocols that you'll be using, and how to ID the target organisms. And a big part of this is feedback. One of the things you'll be doing as you look at your sample is taking photos. And you will be sending those to me via email. So I will go through those and confirm your IDs and get back to you on, yes, this ID was correct, or no, what you thought was this is actually this, and that's why. So we will be developing this back and forth. You will be provided with feedback on how your IDs are. And that's also another important reason, because that's how I QC our data that are entered. So we'll talk more about that as well. We do have an app, if you are unaware, called Phyto. This updated version does include freshwater species now, and we're actually in the process of updating the update. So you will find pictures, pronunciations of the scientific names of these organisms, and if you are using an iPhone or iPad, you do have access to the updated version, and you can even enter your data from your iPhone or iPad. And unfortunately, the Android version has not yet been updated with freshwater. So we are working with our developer to get that going. The gentleman that does the app and support is an IT engineer for a space program. So he's a little busy with his day job. So as soon as that Android version is ready, we will let all of our volunteers know. So what is a HAB? It stands for harmful algal bloom. And this is basically what happens when microscopic organisms can grow very rapidly. And sometimes it's referred to as out of control, but when they have the proper environment, enough light, enough nutrients, they can grow much faster than they normally would. So a lot of times these blooms are not harmful at all. They can be aesthetically unpleasing. They can cause other types of trouble, but they're not technically considered harmful. So think of springtime with plants on land. When there's more light and it gets warmer, they will start to grow again. They kind of come out of their winter dormancy. So that growth rate is pretty normal. But harmful algal blooms are when we have particular organisms that can cause harmful environmental and health effects. So for our program, what we're looking for are certain types of cyanobacteria. Now, cyanobacteria we'll talk more about, but they are true bacterial cells. They are prokaryotic organisms. So they're not true algae. And so even though we still use the term harmful algal bloom, we do stick that term cyano in front to differentiate and to indicate that they are indeed caused by cyanobacteria and not algae. And with our particular program, we are focused on our 5 target organisms. And these targets were selected because they tend to be found in surface water, so it's easy to sample and find them. Although some of them do have mechanisms where they can change their position in the water column. But most of them do have mechanisms to keep them up closer to where the light is so they can photosynthesize. There have been a lot of taxonomic changes over the past decade or so. So you'll see for some of those target names, the also known as. So those are the older taxonomic designations. And the current ones are the ones that we will be using at the top. So we have Dolichospermum, Aphanizomenon, Rhiphidiopsis, that's the most recent taxonomic change, Microcystis, and Planktothrix. So what are cyanobacteria? These used to be referred to as blue-green algae because they were thought to be true algal cells because they could photosynthesize. But what scientists learned is that they are indeed prokaryotic celled organisms. So what I mean by primitive there is that these were the first types of cells to evolve on the planet. that could photosynthesize. Prokaryotic cells do not have a nucleus. They lack a lot of the more complex internal cell structures that eukaryotic celled organisms have. So our cells in our bodies are made of eukaryotic cells. So cyanobacteria are much more simple in their anatomy and construction and processes. But cyanobacteria were the first ones to be able to photosynthesize, meaning they're using solar energy to convert that into glucose, into a food source for themselves. So that's a really important environmental function of cyanobacteria. They are providing oxygen as a byproduct of photosynthesis that can be used by other organisms in the water, and it also gets into the atmosphere for organisms like us to breathe. They also serve as a food source, so they are primary producers in the food web, and so other organisms can feed upon those cells also. Now, when they do grow rapidly, they can form dense blooms when the conditions are right, and that is when we refer to them as a cyanohab. And that picture on the bottom right is Lake Erie, if you're unfamiliar. But you can see they can cover very large geographic scales. So this is an image from 2007, a big bloom that happened then. And they can be quite dense, so sometimes they can form thick scum layers that look like paint, like green paint has been spilled on the surface of the water. But all of that is being caused by these teeny tiny microscopic celled organisms. So what makes a bloom form? There are various environmental parameters that go into making conditions conducive for blooms to happen. But two big ones are nutrients and temperature. So climate change is actually causing more conducive conditions to exist more frequently. So with climate change, we are seeing more storms, more heavy precipitation, and those storm events can wash additional nutrients through runoff into bodies of water. The nutrients are what they need to grow. With increased temperature with climate change, the water temperatures are also increasing. And cyanobacteria love very warm and nutrient-rich water. They also prefer slow moving or still waters. So when you have all of those conditions ripe, that's when you're going to have your tendencies to see these types of organisms grow very rapidly and cause a bloom. Now you can't, unfortunately, just look at a bloom and tell right away if it's a harmful one or not. So, could it be harmful to organisms or animals or even humans? Unfortunately, that's not really possible. There are monitoring programs where you can take pictures of blooms and send them in. But what our program is really more about is determining what are the organisms present and contributing to that bloom. So it's really great to be able to identify these organisms using microscopes to really get an idea of which organisms are present and are those ones that are present potentially harmful. So when we use the term harmful, there are several different ways that that can be applied. So one way that these blooms can be considered harmful is that when those scum layers are very thick, they can block sunlight to submerged aquatic vegetation. And that will lower the overall amount of dissolved oxygen in the water column. So lower oxygen, that's called hypoxia, as you can see on the scale on the left-hand side. When there's no oxygen, that's referred to as anoxia. So those are very dangerous conditions for aquatic organisms. If there's not enough oxygen in the water for them to be able to breathe, that can lead to fish kills. When blooms are present and they eventually die off themselves, there are other bacteria in the water that will degrade those cells. And that process of bacterial degradation also consumes oxygen in the water column. So that can also lead to hypoxic or anoxic conditions. One of the most common ways that people think of harmful, though, is with the production of toxins. And if cells have the genes to produce toxins, and those cells die off and rupture or they're consumed by an organism, that can be a potential harmful health effect. That can affect drinking water, that can affect recreational water. So when humans ingest contaminated water or organisms feed on the cells that contain the toxins, um, it's ingestion. That's the primary way that organisms and animals are exposed to the toxin and thus have the harmful effects. I have family that lives in Salem, Oregon. I visited there going on 3 years now, and they had a water advisory while I was there. So I've experienced toxin in the drinking water firsthand, unfortunately. Most people are familiar with the Toledo, Ohio drinking water contamination event. I believe that was in 2016. Um, so we have had incidences where water sources for drinking have been contaminated by these toxins. Another thing that's been in the news are pets that drink contaminated water. And they're much smaller and they drink much more water than we do if we're out swimming in a recreational area. So they get much more toxin ingestion and can get very sick and can die much more quickly. So they can sometimes, unfortunately, serve as the canary in the coal mine for those types of body of water where there are toxins present. So the toxin can be distributed throughout the food web. And so this is using microcystins as an example of the different ways that the toxins can enter the food web. So again, it's either primarily through drinking the contaminated water or feeding on the cells directly, or even feeding on something that has fed on those cells. So it can actually go up the food chain through different organisms. So it can affect a wide variety of organisms, both aquatic and terrestrial. So the toxins are not a simple, straightforward story in and of themselves. So as you can see from this chart, there are more than just our 5 target organisms that can potentially produce toxins. Our 5 targets are highlighted there, but there are other cyanobacteria that can also produce toxins. The other thing is that you can see that our targets and other cyanobacteria are capable of producing more than one type of toxin. So there's 2 main classes. You have hepatotoxins, and those are toxins that will affect the liver. And you have neurotoxins that will affect the nervous system. And the symptoms for those can overlap with general GI distress. But hepatotoxins tend to be more due to chronic exposure, but neurotoxins will affect the organism much more rapidly. So with the different types of classes of toxins that it can exist, and the fact that some cyanos can make more than one type of toxin is a very complicating factor. So there are various health effects. Again, ingestion is going to be the main route of exposure for humans to become ill. But inhalation is another route of exposure. You can also have what we call contact dermatitis. So you could go swimming in a contaminated body of water and get a rash or skin irritation. So see, these are some of the various symptoms that can be associated with exposure to cyanotoxins. Some are more harmful than others. So again, those neurotoxins are the ones that can really be especially harmful and very quickly. Now, can you tell that toxins are present just by being able to identify what's in the water? Unfortunately, the answer to that is also no. So here you can see a photograph of several different colonies of microcystis. That's one of our target organisms. But you can't tell just by knowing microcystis is there, is it producing toxin? Some of those colonies might be, some of them might not be. So again, it gets complicated very quickly. First of all, not all toxic blooms are discolored. So the water may contain toxins but look absolutely normal. It might not be discolored by the excess number of cells in the water, but there can still be toxin present. And that can be for a variety of reasons. It could be that the toxin producing cells have sunk below the surface and are no longer visible. Or it could be that the cells have died off, but they have ruptured and released their intracellular toxin into the water. So the cells aren't there anymore, but the toxin is still present. Not all species can produce toxins. They need to have the genes. So there are some species or even strains of Microcystis that don't have the genes to produce the toxins at all. Even if they do have the genes, that doesn't mean they always have those genes turned on. So they could possess the ability to produce toxins, but they may or may not be at any given time. And then within the toxins themselves, it's not just one toxin. You don't have one type of neurotoxin. You don't have one type of hepatotoxin. So even with the, again, using this example, the microcystins that are produced by Microcystis, there are over 100 different forms of that toxin. So it's not just one chemical that we're looking for. There are a variety of what we call congeners of that toxin. And the toxicity of those various congeners can vary. Some can be more toxic than others. So you may be at this point be wondering, what's the point if we can't answer all of these questions? It's still really important to know whether or not you have organisms that have the capability, because a lot of the agencies that are responsible for making decisions need to know at least what organisms are present. So that's where we come in. Monitoring is still very important to do, even if we can't answer all of those questions in and of ourselves. We know that harmful algal blooms are increasing, both in their duration, how frequently they're happening, and in their geographic distribution. So again, with climate change, conditions exist earlier in the year than previously, and they extend longer into the year because of those increasing temperatures. With those increasing temperatures, we're seeing organisms cause these harmful blooms in different areas where they hadn't normally, so at higher latitudes and even at higher elevations. So by at least knowing what's present, we can get a better understanding of what conditions are happening and what the potential for harmful health effects are. So we can also work in conjunction with other tools that are available. So if you see in that center picture, that's some satellite data of Lake Erie. So you can see the brightly red areas are where there's a lot of cells present in the water. So that's an indication that a bloom is happening. And what that satellite can't tell you, though, is what are the causative organisms? What organisms are contributing to that bloom? So we can work with those tools to better understand what that bloom is, and they can be put into models to better forecast when conditions might be good for blooms to happen, and thus people are better able to prepare for harmful algal blooms when they do happen. So monitoring really can act as an early warning system for potential toxin exposure. By going out to a recreational area, for example, and monitoring on a regular basis, you can see what organisms are present. Is their abundance increasing to an amount that might be considered harmful if it could be potentially toxic? And so those agencies can go out and put warning signs out to warn the general public that, hey, it might not be a good idea to go swim here. Keep your dogs out of the water. So again, this is where our program comes in to help. There just isn't enough funding, and there's not enough resources for the government or even private institutions to be able to monitor all of the different lakes and reservoirs that are out there that could be potentially impacted by HABs. So by engaging local citizens, In this monitoring, we're increasing the amount of those areas that can be monitored. And again, we can communicate with those agencies that are in charge to make them aware of these blooms that are happening in areas that they might not be unaware of. So PMN started back in 2001. We are now entering our 20th year doing coastal monitoring. But in 2015, we entered into an interagency agreement with the EPA. So normally, NOAA does not get involved beyond the Great Lakes with freshwater. But by engaging with EPA, we have been able to expand this program to monitor freshwater environments beyond the Great Lake region. And what we did is they asked us, can you help us? And we said, sure. And we tweaked what we already had in place to work for freshwater monitoring. And you can see from these two photos, it might not look very impressive, but even our relatively new freshwater program, our volunteers have found blooms, and we were able to confirm 9 of those have been toxic. And that number is actually going to go up because I'm going to be adding a few more for 2020. You'll notice in the marine side, a lot more blooms have been reported, and not as many have been confirmed as toxic events, but it's still really important to maintain the monitoring. So in those events where they do end up being toxin-forming blooms, again, we can alert those agencies so they can make better management decisions. And do things like close down shellfish harvesting areas to the public, or in freshwater, same thing, or again, post those signs to warn people of the potential danger. Okay, so that is the background part. So what we're going to do next is move into what you'll actually be doing when you go get a sample. So you will need to select a site if you have not already. Where you sample is completely up to you, but you need to be able to do it legally and safely, especially in these times of the coronavirus. So please, if you do go out and sample, please make sure that you are able to maintain safe social distancing and wearing masks in case other people come up to you and ask, hey, what are you doing? I just had some folks talk to me about that yesterday as they work on a public pier and they had people coming up to them. So please make sure that where you're sampling and when you're sampling can be done safely. The other major consideration is, can you get to that site on a consistent basis? So the minimum is to be able to sample every other week for 1 year. If you live where it's cold and your pond will freeze, it's ice out to ice in. So it could be something simple as, this is where I have seen blooms happen before. It could be someplace that's important to your community, like a pond in your neighborhood or a lake or someplace where you go swimming. Or you might just be curious to see, what is it in my pond or lake? It's not necessary to go on the same day or at the same time necessarily. The schedule is what's going to, again, work best for you so you can get there on a consistent basis. Generally speaking, these organisms do come up closer to the surface, in the morning when the sunlight is coming up, and they will tend to sink down later in the day. So just kind of keep that in mind. So what I will need from you is a name for your site, just something short and sweet. You can make it up yourself. And I will need the GPS coordinates. So if you have not already done so, please email me that information and I will get you into the data entry portal so then you can start entering your data. So here's the overview. You're going to go out at least once every other week during your sample seasoning. If you can go year round, fabulous, go year round. But otherwise it's ice out to ice in. You're going to take that sample back to your microscope, look for the 5 targets, take pictures as you do so, and when you're done, you're going to enter your data into our database. That last one is going to be on an as-needed basis. So if you see a bloom of something, you will be in very close and very quick communication with me, and we will talk in more detail about what's to be done in that situation. So sampling, you're going to get two different size water bottles, a small 125 ml bottle and a larger 1 liter bottle. You're also going to get a thermometer. Those are what you should take with you to your site every time. We do suggest if you sample the way the gentleman in the photo is that you wear gloves in case you might be susceptible to some contact dermatitis if there is something in the water. And you're basically going to just fill both of those water with the surface water. So you want to, again, select a site where you can get to the water easily. It doesn't have to be a dock or a pier. You can go along a shoreline. I have seen volunteers get very creative with how to extend their ability to collect a sample or maybe go a little deeper. I've seen bottles duct taped to broom handles. I've seen people use the poles that you use for pool skimmers, and they've screwed on bike rack bottles to that, and they put their water bottle in the bike rack bottle holder and get their sample a little further away from the shoreline, or again, a little bit deeper below the surface. But this method is completely acceptable as well. I do also recommend that if you do this method pictured, that you buy the long kitchen gloves that go up to your elbow, because that way you won't get water run down into your wrist and you can get your hand a little deeper below the surface. But we do suggest you wear gloves. You're also going to collect air and water temperatures. Those are our only required environmental parameters to measure. You can make optional weather observations if you do have fun toys to measure things like pH and dissolved oxygen level, or perhaps even a Secchi depth. You can jot those down as well. We love to have that extra information if it's possible. So what you see on the right here is an example of what your data sheet looks like. So you can make copies of those and take them out with you. So you do need to make note of the date and time when you collect your sample as well. You can also jot down what does the water look like? Does it look different than normal? Is it discolored? Does it smell bad? Do you see dead fish floating nearby? Anything that seems out of the ordinary. You can make a note of that, and there's a place to jot that down in the data entry portal as well. So sampling itself doesn't take that long, 5–10 minutes. Ideally, you want to take that sample back to your microscope as soon as you can. But if you know that you've got to go collect your sample and then you've got other things to do and it's going to be a few hours before you can get to the microscope, we ask that you go ahead and preserve that sample on site. And you will be receiving a solution called Lugol's. So this is basically a mix of iodine and vinegar, basically. So this is what you'll add to your smaller bottle only. You'll add 2.5 milliliters. You'll be given a pipette that's marked. So you'll fill it to that top mark with the Lugol's, add it to the smaller sample bottle, and it should look like strong iced tea. If you have a lot of stuff in your sample, you might have to add a little bit more Lugol's to get it to that darker brown color. You will not ever add Lugol's to that 1 liter bottle, because in the event that we ask you to send that sample to us, we will use that bottle for toxin testing. So it can't have anything added to it. So again, if you are going to be holding on to that sample for a few hours before you get to the microscope, you also want to keep the sample bottles out of direct sunlight and heat. So please don't throw those bottles in the back seat of your car and let them bake there while you're running errands or going about whatever you're doing. So again, you might want to bring a small lunch bag cooler with one of those blue refreezable ice packs, and you can keep all of your supplies in there and your samples in there, again, until you can get to the microscope. Once you do get it to the microscope, you are going to be looking for these 5 targets. So these are the ones we're going to train you to ID today. So when you're ready, you're going to take that smaller bottle and gently just mix it back and forth a few times, just to resuspend everything so it's of the general concentration that it was when you took the sample in the water. You will use one of the provided pipettes to take sample out of that bottle, and you're going to add 2 drops to the gridded slides. So you'll be provided with these gridded slides. It's an 8 by 8 grid, and so this limits the amount of area that you need to look at. Excuse me, how'd that go up there? So instead of just wandering around a big blank glass slide, this restricts your area. To make that slide, you're going to add the 2 drops. You'll add your cover slip with one side touching the slide, and you will slowly lower the opposite end. And what that does is that will squish all the air out. So hopefully you will have very few air bubbles in your sample. You're then going to use those grids as a guide and go square by square looking for the target organisms specifically. That's what we're interested in monitoring for. You're going to use predominantly your 10x objective as your scanning objective. So you'll go through one square with your 10x looking for potential cyanobacteria. When you see something you think might be one, you'll go up to your 40x objective, and that's the power that you will use to make your ID and take your picture. We'll talk more about pictures in a second. But you'll then, take your picture, make your ID, go back to your 10x, continue scrolling around grid by grid using the lawnmower method. So you look at all of the grids across, then go down and backwards just like you're mowing the lawn. You also want to be keeping track of generally how often or how frequently you are observing each of the 5 targets as you go through your slide. So not only are you looking for the targets, but you're trying to gauge the relative abundance of each target. So it's a lot to kind of keep track of. Let's talk about taking photos more first. Again, you want to take photos using the highest magnification. That's your 40x objective, which on most microscopes is giving you a 400x total magnification. You will not be using an oil immersion objective. It's not necessary, and it gets really messy. So you should be able to identify our targets using that 40x objective. Now you may need to take multiple photographs of one organism. A lot of these are chain formers, so they can be very long. So it might take 2 pictures to get the entire organism, both the top and the bottom of it. But if you see a target and you identify something as being a target organism, I need at least 1 photograph of that as a representative example of that target group so that I can confirm that ID. Now, you can take multiple pictures of that same target group. For example, if you see multiple colonies of microcystis. I just need at least 1, but feel free to take more. What I don't need is a picture of every single colony of microcystis that you may see. So I just need 1 or 2 representative photos of each one. If you don't have a fancy microscope camera already, you don't have to have one. You can use your cell phone camera. So let me see if this link will work. I will play this quickly. I will provide the link to this video as well. But basically what you're doing is, I think you know, you're going to basically hold your camera with one hand, brace it against the objectives there as she's doing, and then you can hold your phone with your index finger and thumb. And then what you want to do is you want to guide your camera over one of the eyepieces, one of the oculars, and see that circle of light. That's the field of view. And you kind of move it closer until things can come into focus. And then you can use the thumb on your opposite hand to take the picture. Now, one of the hints that she has there is if you go ahead and zoom in a little bit first as she's doing there, what that does is when you take the picture, it gets rid of the black around the field of view. So it's all just light. And again, you can it makes for a much better picture. So that is how you can use your cell phone camera to take a picture with your cell phone. And that's what the majority of our volunteers do. Again, these pictures are really important for me to be able to confirm the IDs for our data quality. And it's also a way for me to provide feedback to you so you know that you're doing a good job. Now, when you do go to send the pictures, what I ask is that you put all of the pictures into one file. So put them all into one Word document or a PowerPoint or make a PDF of them. But you want to make sure that you have each picture labeled. You don't have to label it directly on the picture like this, but maybe next to it with what you think it is. So that way I know what you think it is. You can add the information. I definitely need to know that these photos are from this date at this sample site. So you need to include all of that information so I can always easily equate these pictures with this sample taken on this date by you. You'll be emailing that file to pmn@noaa.gov. So you will not be emailing them directly to me, but that's basically my second email address. I just like to have all of the pictures in one place so my other email doesn't get all gunked up. So me and myself and the director have access to that email address. So that's where we ask that you send the pictures. As you're looking through your slide, again, we're really focused on, are any of those 5 targets present? That's what we really want to know. So starting out, that's the most important thing to look for. There could be tons of other cool stuff in there, and that's great. But really we're focused on, are these 5 targets present and in what abundance? As you get better, you will be able to go through your slides faster. You might be interested in what else is in there. Or you're not sure of what something is. If you're not sure of what something might be, take a picture of that as well, and I'll do my best to try and help you figure out what it is. And there may be some times where you collect samples and there's no targets in there at all. And that happens, and that can be perfectly normal. But even in that situation, you will still need to enter your data. So even if you don't see any targets and you don't take pictures of anything, you still need to enter the data for that sample. Okay. When you've gone through your slide, you are ultimately going to do your data entry. But this is also the part of where I go through, well, how am I keeping track of how many of each thing is present? There is no counting of cells or natural units of these if there are more than one cell. So on your data sheets, you will notice that next to each of your 5 targets, there is a no, yes, or elevated column. So we're not asking you to count them. We're asking you to, as you're going through your slide, kind of keep track of, well, what is the relative abundance of each of these? So what no means is obviously no, I did not see any of that target on the slide. Yes means if you see one cell or one natural unit of a target, that will be a yes. And yes will cover up to 60% of those grids on your slide contain at least one example of that particular target. Elevated is when you start to see more than 60% of the grids containing that target. Remember, you need an answer of no, yes, or elevated for all 5. So that's what we mean by keeping track of relative abundance. It is subjective. I'll have some pictures in a second to give you an idea of where to draw that line between a yes and an elevated. But that's what you're doing as you're looking through your slide. So at the end of your slide, you've gone through all 64 squares on your gridded slide. You basically need an estimate of, are there none, are there some, or are there lots of each of those targets? So what does that look like? Well, again, no, you're not going to see any of those targets on your slide. Again, remember it's individual for each of the 5 targets. You need an answer of no, yes, or elevated. Yes equates to some. So if you see 1, it's a yes. But basically what some or yes translates to is you're seeing that particular target sporadically across the slide. You might see them clumped together, in one section of the slide, but you might not see them anywhere else. But basically, if you are seeing them present in less than 60% of the slides, then that is going to be a yes. Elevated, one will usually be pretty obvious. You will see multiple examples of that target in more than 60% of the grid. So that means you'll be seeing 4 or 5 in 60% or more of your gridded slides, or the gridded areas on your slide. So it's going to be the dominant organism on your slide. And sometimes it will be the only organism you see on your slide, and that's pretty much the bloom situation. So you can have blooms where you have still some, species diversity present, but again, your target is going to be the predominant organism across the slide. So where to draw the line between a high yes and a low elevated, again, that's subjective, but the more you do this, the more you'll get a better idea of what that is. So it's really when it's elevated, that's when we are concerned. So that's when the communications start, is when you see elevated levels of one of those targets. You're going to email me immediately and let me know, and then we will discuss what to do from that point on. So that's why I don't go into great detail of what do I do when I see lots of elevated things. What you do is you email me or you call me. That is the protocol. Ultimately, though, again, you're going to enter the data on our website. So you will go to our homepage, and there's an area that says Submit Data. You will click that, and you will see a list of all of our different regions for both programs. But here is freshwater, and that is where you will click, and that will bring you to the page. You will fill out your name and your email address, and you will enter all of your information. And it's not going to open for me. Okay. But it's just a Google form. It's very straightforward. And you're going to enter all of the information that you see on your data sheet. So you'll have a no, yes, or elevated for each of your targets. You're going to enter, you'll select your site number that will already be in there for you to choose from, the date, the time, the water temperature, and the air temperature. Air temperature is required. It's actually up here now. Any of this optional information you can enter. What other types of organisms you may have observed, that's all pre-filled in as not applicable for you. But if you can identify that something is a diatom, go ahead. If you can identify other cyanobacteria, there's also a list of some of those to choose from. But please keep in mind, if you identify something by name, I need a picture so I can confirm that identification. So again, the symbols here, I forgot to mention this. You'll notice there are symbols in the no, yes, or elevated. So circles mean don't do anything. You don't need to call. That's fine. You will enter your data and send me your pictures if you have any. What you'll do in those situations is you will empty both of your bottles, rinse them out, reuse them. So when you go out to collect your sample with those bottles again, you'll basically dunk them 3 times, just so fill them up with a little bit of water, shake them, rinse them out, dump it. Do that 3 times for each bottle, and then collect your new sample in those bottles that you're reusing. Again, if you see elevated levels of something, that's the little diamond. Down here it tells you contact PMN staff, and we will talk about how to maybe ship the samples if that is determined to be needed. So enter your data even if you do not see any targets for that particular sample. Now, what to do if you do have an obvious bloom? Yes, you are going to, again, contact us first to confirm the ID of that organism. And then you and I, or together, will contact the state or local regulatory agency that is in charge of harmful algal blooms and reporting them. So, we can contact them and see if they want the sample shipped to them for toxin testing, because again, we are very limited in our ability to do toxin testings. So, they'll perhaps tell us the shipping protocols, and we do appreciate if they get back to you and let you know, or if you are the actual entity doing the testing, If you could please just share that information with us so we can keep track. That toxin information is not shared on our database at all. That is something that we keep track of internally only. Again, we do have, unfortunately, limited capability to test for toxins. Right now, we can only test for microcystins using an ELISA kit, and it has to be done when we have enough samples to warrant using those kits. They're pretty expensive, so we can't just run one sample at a time on them. So that means the results are not going to be immediately available. So it really is best to go through the regulatory agency. If it's the Department of Health or Department of Water or Department of Wildlife, whatever the state agency is. So of the 5 targets, 3 of our targets are known to be potential microcystins producers. So that's Dolichospermum microcystis and Planktothrix. So if you were to see elevated levels of those and your state agency doesn't want the sample or can't take the sample or can't do toxin testing, we may ask you in that instance to then send the sample to us. So just a brief review of what you're doing. You're going out, collecting that sample every other week, looking for the targets, taking pictures as you go, and entering your data. Okay, here comes the fun part. This is where we're going to go through how to ID these things. So we've talked about how to collect the sample, but how do you actually know what Dolichospermum is? So again, these are cyanobacteria that we're looking for. They are small. They are, again, true bacterial cells. They have evolved over billions of years to live in a wide variety of environments. So they're in fresh water, they're in brackish water, they're in marine water. And they can take a variety of forms. They do exist as unicellular organisms, but some may form what we call multicellular natural units because when the cells divide, they stay stuck together. They're also colonial forms. So again, these very small microscopic organisms can have huge effects and cover huge areas. So what is the actual difference between true algae and cyanobacteria? When you're looking at things on your slide, how are you going to be able to tell, oh, that's really an algal cell versus, oh, that's a cyanobacterial cell, especially if it's just one cell by itself? Well, algae are eukaryotic celled organisms. They're going to generally be bigger in size than the individual cells of cyanobacteria. So eukaryotic celled organisms have, again, a more complex internal structure. They do have true nuclei. They have membrane-bound organelles like pyrenoids, which store starch. So they will have these, noticeable, for the most part, internal structure elements that are well defined. And you can say, aha, that must be a eukaryotic cell. Sometimes they're really cool. So this bottom one, Spirogyra, the spiral chloroplasts are a dead giveaway. That is a eukaryotic celled green algae. These unicellular ones, you can see the pyrenoids very easily. That is a unicellular true algae. Cyanobacteria are going to be much smaller in size individually. Now, when they do form these longer chains, they can be quite large overall in size, but the individual cell size is still very small. They will not have nuclei. They may have some internal structure to them, but they're going to be very small, like tiny dot-like structures, like these starch granules here in that top middle picture labeled B. So that's really the best way to tell the difference. Am I looking at something that's a cyanobacterial cell or a green algal cell? Is it still difficult sometimes? Absolutely. So one of the things I have learned over the years and being trained by other professionals in this field that have been doing it way longer than I have is that it is not always possible to identify every single thing on your slide. So that was very helpful for me because I'm the type of person that wants to be able to identify everything. So even when you send me your pictures, I may not be able to tell you exactly what's there. But as you again, practice, it's going to get a lot easier to tell the difference. So here are some of the terms I'm going to be using as we go through our 5 targets, looking for particular features that can help you characterize something as an individual target. So most cells just exist in their day-to-day being as what we call a vegetative cell. So this is just a normal cyanobacterial cell doing its thing. Some cyanobacteria do have specialized membranes within them that are called gas vesicles or aerotopes. So these are membranes that will have gas in them, and this is one of those mechanisms that some have to maintain their buoyancy in the water column. So they can increase the amount of gas in those aerotopes, and that floats them up closer to the surface so they can take advantage of the sunlight to photosynthesize. They can then collapse those aerotopes, and that will cause them to sink deeper in the water column. So cyanobacteria that do have aerotopes, not all of them do, are able to regulate where they are in the water column very, very well. So what aerotopes will make vegetative cells look like is sort of modeled. So most of you are probably using regular light microscopes in what we call bright field. So what those aerotopes will look like are dark spots within the cell. So it'll give the cells sort of an overall modeled look. So that's what that's one feature that you'll be looking for. An akinete is when 2 or more vegetative cells fuse together, and they will have this extra thick wall around them. So they will become much bigger and much thicker walled than any of the vegetative cells. And what akinetes are basically a survival mechanism. They act as resting spores. And what that means is, for example, it might be fall, water temperatures are dropping, and what they'll do is they'll form these akinetes, and all of the vegetative cells will die off, but that akinete will sink to the sediments and overwinter. And they could overwinter and come back and emerge as more vegetative cells in the springtime. They might stay submerged for decades before they reemerge. So they can actually survive very long periods of time. So again, what you're looking for to distinguish something as an akinete is they will be much larger than the regular vegetative cells. Now another special trait that some cyanobacteria have are these heterocytes. The old term is heterocyst. Those are the same thing. So heterocytes are what cyanobacteria have if they are able to fix nitrogen. So that is a specialized process that some cyanobacteria can do that can take atmospheric nitrogen out of the water column and turn it into bioavailable forms of nitrogen. Nitrogen is a very important molecule for protein formation, which is a very important part of cell anatomy. So having nitrogen available is a really important growth requirement. So that's where you hear about excess nitrogen in the waters, making cyanobacteria very, very happy. Some of them can fix nitrogen themselves. So in the event there are lower nitrogen levels available in water columns, that kind of gives them an advantage over other types of organisms that can't fix nitrogen. So that's an important process. If they can do that, a heterocyte will look, again, much different than a vegetative cell. They'll generally still be the same size, but notice how it looks empty now. There's no coloration. And it also has a thicker wall around it. So it kind of almost has a halo effect to it. So those can be very distinguishing features that can help you identify certain types of cyanobacteria. Again, if you see these features, those are things you want to take pictures of. So that helps a lot. Now, will they always have heterocytes or akinetes? No. Again, that's why you have to maybe keep looking for some that do have these more easily identifiable characteristics to make a good ID. So again, you might not be able to identify every single thing in your sample, but you're looking for the ones that you definitely can. A lot of our targets will form what we call trichomes. Some people say trichomes. It's fine. Tomato, tomato. But trichome refers to a row of cells that are connected. So when these cells go to divide, they will stay stuck together and can form these long filaments. So that's what we refer to as a trichome. Trichomes can be unbranched, meaning they don't have offshoots. It's just one length of cells. They can also be tapered or untapered. So tapering refers to a narrowing of the cell or a reduction in size of the cells towards the ends of the trichomes. So if they're untapered, the cells at the end will be generally the same size as all of the other cells. If they taper, they will get noticeably smaller, narrower in that size. So it's also important to take pictures of the ends of your trichomes. So again, you can see in this upper picture here, you can see really cool features like heterocytes and aconites, but you can't see the ends of either one of those trichomes. So you might need to move your slide a little bit and then take another picture and maybe put both those pictures on the same page or next to each other so that I know these are the same thing. Now, some cyanobacteria can make these extracellular coatings called a sheath. So this is what I equate to, putting a worm into a straw. Okay, so the straw is the sheath. And so this is outer covering of the entire trichome by this extracellular matrix. And not all cyanos do that. None of our targets will have a sheath. But that's what they look like. So you can see it's definitely outside each of the cells and even extends beyond the trichome itself. Okay, so here we go. Target number 1 is Dolichospermum. This used to be called Anabina, but Dolichospermum are basically Anabina that have aerotopes. So Anabina still exists, but Anabina do not have aerotopes. So Dolichospermum is a filamentous form. Those trichomes are going to be unbranched and untapered. And they usually exist as solitary trichomes, meaning that they don't associate in any formal way with other trichomes of Dolichospermum. They do not have a sheath. I will explain the asterisks shortly. There are lots of different species of Dolichospermum. Some will make a straight trichome, some will be curved or coiled, and some will actually be very symmetrical helices. So there's a lot of variety in Dolichospermum, but they will have these traits in common. So what you're looking for, and these slides, let me just point out, are basically the pictures that are in that Cyanobacteria ID guide that you were emailed as well. So that's what I'm going through here. So what you're looking for Dolichospermum, the individual cells are going to be round or barrel shaped. You notice that they look mottled. So these darker spots within this picture, those are the aerotopes. So that's what gives them that mottled look. So those indicate aerotopes are present. If they have heterocytes, those are going to be in what's called the intercalary position. So that means they're going to be somewhere along the middle of the trichome. They're not going to be found at the ends of the trichomes. Likewise, aconites, they will be in the intercalary position as well. And one really great feature of Dolichospermum is you can see the individual cells very clearly because the cells are constricted where the cells meet at their cross walls. So it almost makes Dolichospermum look like beads on a chain. So that's very distinguishing of Dolichospermum. Are there others that have cell constrictions like that? Yes. So again, you're looking for all of these things together to identify it as Dolichospermum. So again, you're looking for aconites. The aconites are going to be bigger than the vegetative cells. They sometimes look like beans or kidney beans in some species of Dolichospermum, but they will be noticeable. Heterocytes, again, they're going to be the clear ones. And again, in this picture here, you can see that this trichome, it might actually be two trichomes. You can see some of these features in focus. So again, you want to have a focus picture of an aconite, a focus picture of a heterocyte, but everything else is kind of blurry. So again, you might have to take more than one picture of this colony. So you can't see the end. So I can tell right here in the upper left, that's the end of one of the trichomes. So I might focus through a little bit more to take a picture of that end cell, or maybe over here to get that in focus, to get a good look at what the end or terminal cells look like as well. So those are the features you're looking for. Here are some other pictures of Dolichospermum, kind of showing you the variety of the cells, the shape of the aconites. Some of the pictures you'll see through this presentation are either stained cells, so they'll all look dark. That's what your cells will look like if you have stained them with your Lugol's. Others use special filters that you might not have for your microscope. So that's why they all look a little bit different. This picture on the bottom left is the one that has to do with that sheath and the asterisk next to it. So you can kind of see this watery outline surrounding this trichome of Dolichospermum. Some will say that is a sheath, some say it's not. I don't have a dog in that fight, so I just put the asterisk next to it. Now, sometimes it can be confused with another cyano called nostoc. The cell shape is very similar. Nostoc also has heterocytes and aconites, but what they don't have are erytopes. You'll see that they lack that more mottled look. And they're also colonial. So nostoc will be encapsulated within this gelatinous matrix. Okay, so it's like a big snot ball. And all the trichomes will be within that capsule. So nostoc is technically a colony of trichomes, whereas Dolichospermum, they exist as individual trichomes. Target number 2, a phanosomenon. That one's just fun to say out loud. This is another filamentous form. The trichomes are straight and unbranched, but these do taper at both ends. So here is, again, an important time to take pictures of the ends of the trichomes so you can see how they will get a little bit narrower and change their shape a little bit. They do not have a sheath. And here trichomes do tend to associate with other trichomes of a phanosomenon. They will kind of bundle together in these parallel layers as you see in the photographs here. So that can be a distinguishing trait of a phanosomenon. They do have heterocytes, so again, those clearer looking cells. They can form aconites, so again, the fusing of vegetative cells into these larger spores. And again, they can be long-lasting in sediments. So for a phanosomenon, what to look for? You're looking for the cell shape. So here, the cell shape is more cylindrical. They're not rounded or beads or barrel shaped like Dolichospermum, but they're cylindrical. So think of a soda can. The cells are going to be longer than they are wide, and you're looking at these soda cans from the side, so they almost look more like bricks, but just do know that they are cylindrical in shape. They will be longer than they are wide. Erytopes, when they're present, erytopes are present, so facultative means sometimes they have them, sometimes they don't. Sometimes it depends on the maturity of the trichomes, the cells in the trichomes. But generally speaking, a phanosomenon does have erytopes. Heterocytes are intercalary. Aconites are also intercalary, and instead of being bean-shaped or kidney-shaped, they are just like bigger cylinders. Terminal ends are very, very important to look at for a phanosomenon. So if you look at this trichome down here, you can see the cells. You can kind of still see the cell walls, but as you get closer to the end, look at what's happening to the cells. The cells are becoming even longer than the other vegetative cells, and they'll be rounded, and sometimes they will start to look empty looking. So that is a distinctive trait of a phanosomenon. So some species will get these longer cells. They may taper. Sometimes the ends may actually inflate a little bit. So you can see it gets ever so slightly bulbous at this very last cell, but they can look empty. So those are important things to look for to ID something as a phanosomenon. So again, some other pictures there. You see that rafting together of the trichomes. They don't always necessarily do that. So you can still identify individual trichomes as being a phanosomenon. But in environmental conditions where they're super happy, they tend to raft together like that. Now it can be confused very easily with another one of our targets called Planktothrix. We haven't gotten to that yet, but we will explain more why they look similar to each other when we get to Planktothrix. So in a bloom, again, you can see the bundling together of the trichomes very well. And up close, again, you can see how the cells at the ends get a little bit longer than the regular vegetative cells and more empty looking. And that's what it would look like in a bloom. Please don't stick your hand into water that looks like that without a glove on. Target 3 is Raphidiopsis, another filamentous unbranched form. Again, they're pretty straight, but they can be curved or coiled back to being solitary trichomes. They do not interact or raft together. They might get tangled up with each other, but there's no formal association between the trichomes. They do not have a sheath. Here, again, looking at the ends is where you're going to have to do. Because if heterocytes are present, they are always at the terminal position. They are on the ends. So these flame-shaped or teardrop-shaped structures on the ends of the trichomes, those are the heterocytes. If akinets are present, they will form directly behind or one or two cells back from the heterocytes. So they will not be in the intercalary position. So things to look for Raphidiopsis. The cells are still cylindrical, like a phanosomenon. So the cells are going to be longer than they are wide. They will have aerotopes. The heterocytes, when present, will be at one or both ends. And again, they have that kind of flame-like shape to them. Akinets, if they're present, will be one or two cells back. And if they lack heterocytes altogether, the terminal end cells will be either narrowed and conical, so they're a little bit narrower and rounded at the end, or they can be super pointy. So here on the bottom left is an example of Raphidiopsis that does not have any aerotopes present. But in these other photos here that you can see, again, there's various dyes or filters that are being used in these pictures, but on this bottom one, here you see the heterocyte on one end and not the other. Here again, an aerotope in the middle. And up here is a very distinctive aerotope on one end and not the other. It used to be confused with Raphidiopsis, but it used to be called Cylindrospermopsis, but now they know these have been designated to be in the same genus now. So now you can't confuse them with each other anymore, which is nice. Target 4 is Microcystis. This is probably the one most people have already heard of because this is the one that gets in the news a lot. This is our one target that is colonial in form, meaning that the individual cells are held together in a colony by this mucilage, this mucus. So again, it's a big snot ball, basically. The colonies have no set shape. So they are three-dimensional, meaning that you have cells behind cells behind cells. They're not always hollow balls of cells. But they can have these hollow spaces within the colony itself. They are very buoyant. They do have aerotopes. They can regulate their position very well in the water column. They are apparently very stinky. I've never been in the presence of a Microcystis bloom personally, but I do hear they reek. So what to look for Microcystis? The individual cells in the colony. It's important to look at the cell shape. There are other colonial cyanobacteria, but Microcystis cells will be rounded. They might be a little bit oblong, but they're generally very round. Lots of aerotopes. And again, the colony shape has no set form, but the cells can range in how they are associated within that colony. So they can be very loosely associated, and it can be very difficult to see the colony, the mucus that holds that colony together. Um, Sometimes when colonies are very old and mature, they will take on this clathrate form in the middle. They have these big jagged holes torn in them. And they can be densely packed, so they can have very few um, spaces with air amongst the cells within the colony. And you can sometimes very easily see the mucus holding the colony together. So that mucus can vary in thickness from different types of microcystis, um, making that colony hold together. So here's two good examples of how different they can look. So here is a microcystis aeruginosa on the top, so that's more loosely associated cells. I believe this one on the bottom is microcystis viridis. Tends to be a little bit more symmetrical, but you can see, again, the edge of the mucus holding those cells together. Now, what it can be very easily confused with is another colonial cyano called Waranachinia. Waranachinia makes up a hollow ball of cells. It's not necessarily always a big round hollow ball of cells, but you basically have one layer of cells. Microcystis does not do that. The cell shape of Waranachinia is different. They're actually more rod-like and not rounded. So that's why it's important to look carefully at the cell shape and not just assume every green mass of little tiny cells that you see is automatically microcystis. So this is what a bloom can look like of microcystis, and those colonies will tend to stick together. Again, here's an example of those kitchen gloves I was talking about. So you can stick your hand a little deeper in the water when collecting your sample. Planktothrix is our last target. So previously was Oscillatoria. Oscillatoria still exists, but they do not have aerotopes. Planktothrix has aerotopes. So back to the filamentous form, unbranched. These tend to be pretty straight. So they're not curvy, it's not as coiled as some of the others can be. They'll be pretty straight, or they might be a little bit wavy along their length, especially towards the terminal ends. No sheath, no heterocytes, no aconites. So you will not see any heterocytes or any aconites ever in Planktothrix. So if you see something that you think is Planktothrix and it has a heterocyte, it is not Planktothrix. These might be seen gliding across your slide if your slide is pretty fresh and the cells are still alive. Again, that's a great reason why to get your samples to your microscope quickly, is that you can see them in their true coloration and you can actually see them sometimes moving. Look carefully at the cell shape. They are still cylinders, but now those cylinders are wider than they are tall. And sometimes they're much wider than they are tall, the individual cells making up the trichome. So notice that's the opposite of Aphanizomenon and Raphidiopsis. They are longer than they are wide. So that right there, again, another important reason to look at the individual cell shapes. They can be what's called isodiametric, so that means they're almost the same height and width. They might be like little squares. But what they won't be is longer than they are wide. So lots of aerotopes in Planktothrix. The terminal ends will be rounded if there are a more isodiametric shaped species. The cells will be just gently rounded on the ends. If they are a much wider than tall species of Planktothrix, again, they're just kind of rounded towards the ends. So you will not see heterocytes or aconites. And this can be very easily confused with not Cylindrospermopsis, Raphidiopsis, even I say the old names, because if you have Raphidiopsis that does not have a heterocyte on either end, that can be very easily confused with a Planktothrix that also will not have any heterocytes ever. So again, it might not be able to identify each individual trichome of everything on your slide, but if you see something that looks like this, but the cells are longer than they are wide and they taper at the end and they might get empty looking at the end, you might be looking at a phanosomenon. How to clear my throat. Okay, so some pictures on the left of Planktothrix, and then a picture on the right with something it can sometimes be confused with, is Lingbia. Lingbia is actually a benthic species, but they can break off of their benthic mats and float up to the surface. But usually Lingbia you will see within their sheath. So this is the picture we looked at earlier. Um, so the cells are similar. They're very coin-like. So they're very, very wide and thin, not very tall, that is. And Planktothrix tends not to be quite as wide and thin as that. So again, there'll be filamentous. Again, don't pick this stuff up with your bare hands. There is one species called Planktothrix rubescens, which has this kind of reddish color to it. We actually did get a sample of that last year. It's the first time I'd seen that, so it was pretty cool. So that is the ID portion. So again, thank NOAA and EPA for the funding. If beyond this training session right now you have questions, you can always email me at any time. And so what we're going to do next is do a little bit of practice. So we'll take a quick 5-minute break. It's 10:25. If you come back at 10:30 and ready to go with Menti, or we don't have to take a break if everyone's ready to go. If you want to just type in the chat box if you vote break or no